Welcome to Radicle Health, Introduction to the Endocannabinoid System. This module is intended for medical practitioners, but can also benefit other audiences interested in learning about how cannabis can help treat chronic and age-related illness. A full suite of Radicle Health training modules are available, and in addition to the information presented in this module, they include information about cannabinoids, terpenes, routes of administration, extraction methods, and dosing guidelines. Additionally, disease-specific modules can help you understand considerations when recommending cannabis to patients with a specific condition. A module focused on geriatrics can help you use cannabis to treat illnesses and conditions typical of senior patients. For a comprehensive description of the endocannabinoid system, including descriptions of cell receptors, enzymes, ligands, and drug transporters relevant to cannabinoid pharmacodynamics, and a review of the absorption, metabolism, distribution, and excretion of cannabinoids, see Radicle Health Cannabis Pharmacology. This module provides an overview of the endocannabinoid system. Let's get started. This is a partial list of conditions or disease for which cannabis has demonstrated efficacy in either animal models or in clinical studies. Notice that the list contains examples of neurological disorders, psychological disorders, gastrointestinal disease, infectious disease, and inflammatory disease. This is an incredibly wide range of conditions that cannabis helps treat. When you see this list, your first response should be skepticism. How can one plant help with so many conditions and have so few side effects? To understand the answer, you must understand the endocannabinoid system. Living organisms regulate their internal environment to maintain the relatively narrow range of conditions required for cell function. For example, your body temperature needs to be kept relatively close to 98.6 degrees Fahrenheit. Your pH must be kept within a very narrow range to survive and function. Your blood must approximate specific levels of systolic and diastolic pressures to remain healthy. This maintenance of a stable internal environment, even in the face of a changing external environment, is known as homeostasis. The endogenous cannabinoid system, or endocannabinoid system, the ECS, is our physiologic system required for maintaining homeostasis. In fact, there is evidence of an ECS present in all mammals, in fish, reptiles, earthworms, leeches, amphibians, birds, every animal except insects. The scientific community estimates that the ECS first began to evolve over 600 million years ago. The endocannabinoid system is a physiologic system that includes molecules that our bodies create on demand, endogenous lipids called endocannabinoids, the cell receptors that these cannabinoids bind with, the enzymes that metabolize or degrade the cannabinoids, and neurons and neural pathways where the molecules, enzymes, and receptors are all co-located. The human body includes more than 37 trillion cells. Each cell has a specific function, a specific task or set of tasks that it performs. To achieve these tasks, cells have a mechanism for letting things into and pushing things out of the outer cell membrane. Receptors, embedded in cell membranes, act as gates. When stimulated, the receptors can initiate a variety of physiologic processes. There exist multiple receptors that are modulated by the ECS, but the most well-studied cannabinoid receptors are CB1 and CB2. These are G protein-coupled receptors that are located throughout the human body, including in the brain, organs, connective tissues, glands, and immune cells. Endocannabinoid receptors are the most common type of cell receptor in the body. In fact, there are more CB1 receptors in the brain than any other type of G protein-coupled receptor. The highest densities of CB1 receptors appear in the hippocampus, cerebral cortex, cerebellum, amygdaloid nucleus, and basal ganglia. The homeostatic effects produced by CB1 stimulation include an increased drive to sleep and eat, a reduction of perceived pain, fear, and anxiety, a maintenance of well-being, and the promotion of recovery during stress. There are very few CB1 receptors in the brainstem or in the cardiorespiratory centers, which accounts for the absence of a lethal dose with cannabis. While CB2 receptors are also located in the brain and in the peripheral nervous system, they are concentrated in the peripheral immune cells. for example in the bone marrow, thymus, spleen, and tonsils. CB2 receptors are also located in the uterus, lungs, microglia, and brainstem neurons. The number of available CB2 receptors increases significantly during inflammation. CB2 receptor activation does not produce the psychoactivity associated with CB1 receptor activation. The homeostatic effect produced by CB2 stimulation is mostly the reduction of inflammation. The discovery of the first cannabinoid receptor by Alan Howlett and William Devane in 1988 led researchers to assume the existence of a naturally occurring substance in the body that would bind to the receptor. The first endogenous cannabinoid was discovered in 1992 by Raphael Mechoulam in collaboration with William Devane and Lumir Hannes. Endocannabinoids are the substances our bodies naturally make to stimulate the CB1 and CB2 receptors. The two most well understood of these molecules are called anandamide, or AEA, and 2-arachidonylglycerol, or 2-AG. These molecules have a local effect, are short-lived, and are degraded by the enzymes fatty acid amide hydrolase, or FA, and monoacylglycerol lipase. Our bodies contain nervous tissue, which is made up of nerve cells. One specialized type of nerve cell is called a neuron. Neurons transmit nerve signals through the body and to and from the brain. They communicate by sending chemical messages called neurotransmitters, which help coordinate and regulate how we feel, think, and what we do. Most neurotransmitters travel in a single direction, from an originating neuron, the presynaptic cell, across a small gap called the synapse, and then attach to specific receptors located on a nearby neuron, the postsynaptic cell. The neurotransmitter activates the receiving neuron to initiate the actions required to pass the message to the next neuron. The ECS communicates its messages, the endocannabinoids, in the opposite direction, from a postsynaptic neuron to a presynaptic neuron. When the postsynaptic neuron is activated, fat cells present in the neuron create endocannabinoids, which are released from the cell and which travel backwards to the presynaptic neuron, where they attach to the endocannabinoid receptors. Because endocannabinoids act on cells where neurotransmitter signaling originates, they can limit the release of neurotransmitters and influence how messages are sent, received, and processed by the cell. This type of signaling is called retrograde signaling. Retrograde signaling is the body's feedback mechanism. For example, retrograde signaling can suppress the release of neurotransmitters when a nerve is firing too frequently. This means that a stimulation of the ECS can reduce inflammation, relax muscles, lower blood pressure, dilate bronchial passages, and normalize overstimulated nerves. And, because endocannabinoids are located at the intersection of many physical systems, they enable the communication and coordination among different cell types. For example, endocannabinoids at the site of an injury can minimize pain and damage by decreasing the release of activators and sensitizers from the injured tissue, by preventing excessive firing in the nerve cell, and by calming immune cells to inhibit inflammatory responses. And while the ECS performs different tasks in different types of tissue, its overall function is to achieve homeostasis, the maintenance of a stable internal environment. For example, the ECS mediates the process of autophagy, where a cell isolates, digests, and recycles its own intercellular material. This process causes malignant tumor cells to consume themselves via programmed cellular death. Since the discovery of the ECS in the early 1990s, we've subsequently learned that the ECS is responsible for regulating our mood, appetite, pain sensation, memory, and sleep. Many diseases are characterized by a chemical deficit. Patients and healthcare professionals treating these diseases will administer medicines that supplement the deficit. For example, healthcare professionals treating Parkinson's will attempt to supplement a dopamine deficit with the medication L-Dopa, an amino acid that is a biological precursor to dopamine. And patients with diabetes must inject insulin into the fat under their skin, where it eventually makes its way into the circulatory system. When the ECS is functioning normally, we experience a normal mental state, no pain, and good digestive function. Consider, however, patients who exhibit symptoms disproportionate to any observable cause. These patients suffer from hyperalgesic syndromes, where they feel pain even though the corresponding tissues appear healthy. Patients suffering from migraines, irritable bowel syndrome, and fibromyalgia all exhibit these symptoms. These types of conditions tend to occur in the same individuals. Patients with chronic migraine are more likely to suffer from fibromyalgia and with irritable bowel syndrome. What these patients might have in common is an endocannabinoid deficiency, a baseline of endocannabinoids that is chronically below a healthy level. Dr. Ethan Rousseau originally coined the term clinical endocannabinoid deficiency, and his research has proven to be the foundation for our understanding of this category of disease. Data confirms that the patients with these conditions express lower than normal levels of anandamide in the brain, gut, and musculoskeletal system, and these disorders often benefit from cannabinoid treatment. If we can supplement deficiencies of endocannabinoid levels, then patients suffering from allodynia, fibromyalgia, migraine, IBS, and other related conditions might achieve relief with cannabis. Patients can also suffer from disease when their endocannabinoid tone is chronically raised above a healthy level. Consider patients who suffer from morbid obesity. These patients experience metabolic syndrome with increased inflammation, insulin resistance, and often diabetes. In this scenario, we can expect the ECS to be hyperactive, where the endocannabinoid tone is elevated above normal levels. Similarly, excess of endocannabinoid activity, especially at the CB1 receptor, is associated with liver fibrosis. Stress seems to play a major role in clinical endocannabinoid deficiency. Research in animal models has demonstrated that stress-induced anxiety is directly related to anandamide deficiency in mice, and that increasing anandamide levels reduces this anxiety. In addition to chronic stress, sleep deprivation, chronic alcohol use, and binge drinking can all decrease endocannabinoid signaling activity. Clinical endocannabinoid deficiency can also arise from genetic or congenital issues or from intercurrent injury or disease. There are many modalities helpful for upregulating the ECS. A proper diet, exercise, stress management, massage, and acupuncture can all positively influence endocannabinoid levels. Pharmaceuticals, such as analgesics, anti-inflammatories, antidepressants, antipsychotics, and anticonvulsants, also influence endocannabinoid signaling. The wide distribution of cannabinoid receptors in the body is the reason why the ECS seems to play such an important modulatory role, and influencing ECS activity may have therapeutic potential in almost all diseases affecting humans. In fact, there are very few physiologic systems in and conditions of the body with which endocannabinoids do not participate. This is a partial list of systems and conditions of the body that the ECS does modulate. You can review this list in the transcript. The endocannabinoid system helps us relax, eat, sleep, and forget, and it works to protect our bodies. The ECS is responsible for regulating a large number of systems and conditions. And, because cannabis can help modulate the ECS, cannabis can be an effective and safe option when treating a wide array of conditions and disease. This concludes this module. Contact Radicle Health for more information about the full suite of modules that are available to help you use cannabis to treat patients suffering from age-related and chronic illness.